Assalamu alaikum. I am presenting this presentation on behalf of my beloved and respected supervisor, Dr. Sehrish Faryal, Associate Professor at IBBT, US, Lahore. The topic of my presentation is genomic analysis of single nucleotide polymorphism in complement component 5 gene for mastitis in dairy animals. As we all know that cattle is a significant modern domesticated member. It belongs to the subfamily Bovini, and it is the most common species of the genus Bos, and it is most frequently classified collectively as Bos taurus. Cattle are used as beef and draught animals. They exhibit about 53% of Pakistan livestock population and contribute nearly 35% to the entire production of milk in the country. Pakistan is positioned fourth in the production of milk worldwide, whereas Pakistan's cattle population is recorded at 29.6 million. In our study, we are using complement component 5. This is basically a protein which is encoded by the C5 gene. It is the fifth component of complement system, and it plays an important role in inflammatory and cell killing processes. This gene is cleaved into two regions, that is C5a and C5b. C5a plays an important role in chemotaxis, whereas C5b forms the first part of the complement membrane attack complex. The figure below shows the complete C5 gene with its different regions, and it is located on the chromosome 8. The objective of the current study are to characterize the C5 gene in cattle, as well as to find out the molecular markers in the C5 gene for mastitis susceptible animals. Material methods are divided into three phases. The first one is the sampling, which includes the selection of cows and the collection of blood samples. After that, the experimental work will be performed, which involved the RNA isolation. After that, the cDNA will be was synthesized. The primer were designed along with the PCR optimization. After that, the nanodrop was performed, and ultimately the DNA sequencing was done. The analysis was done by using different bioinformatic tools. So first of all, we selected the cows. 20 Sahiwal cows were chosen for the study, which includes the 10 diseased and 10 control samples. 2 ml of blood was collected in vial containing EDTA, and the blood was stored at -1 degree Celsius. After that, the RNA extraction was performed from the blood samples by using triazole reagent, and the samples were then stored at -20 degrees Celsius. The quantification was done by using agarose gel electrophoresis and the nanodrop spectrophotometers. The extracted RNA was then subjected to cDNA synthesis by using reverse transcriptase, and again the quantification was performed using nanodrop spectrophotometers. 3 sets of primers were designed by using Primer 3 software online, and amplification of exons 6, 8, and 10 of the C5 gene was done by using PCR. The precipitated PCR products were subjected to sequencing by dideoxy chain termination method using ABI genetic analyzer. The last step was a bioinformatic analysis, which includes different software such as BLAST was used for finding out the mutation or polymorphisms. Cluster W was used for alignments, and Mega 7 was utilized for the phylogenetic analysis. All the work was done in the research laboratory of the Institute of Biochemistry and Biotechnology, US, Lahore. Come towards the results. These are the gel results of the 3 exons, that is exon 8, 6, and 10, respectively. Come towards the polymorphisms, we have found that one single nucleotide polymorphism was found at position 113, where G was replaced by T, and it results in the transversion, where we can see that serine was replaced by alanine. These are the phylogenetic results. So at the end, we can say that there is a genetic marker in the C5 gene for mastitis in our population of Sahiwal cattle. Genetic marker GT mutation at 113 position results in a missense mutation in amino acid sequence, where serine was changed to alanine. And it was reported that it can be used as genetic marker for the selection of mastitis resistance and susceptible cattle. Um, the results can be useful for the selection of mastitis resistant and susceptible cows. They can also be used in selective breeding of cows, and also they can lead to better understanding of pathogenesis and progression of mastitis. So that's all. Thank you.